The following notes are in Chapter 10, Section 2 of your textbook on mitosis and the cell cycle. This information follows the information we've covered on DNA and DNA replication. DNA replication, if you remember, is the copying of DNA. And at the end of replication, you would have two sets of identical DNA strands in the nucleus of the cell. So these DNA strands would be in the nucleus. You wouldn't really be able to see them, but you would just know that the nuclear composition would be double. And the next logical thing that would happen is the cell does that in preparation to divide. And that's what our notes are about today. I also wanted to remind you that when we talked about the cell theory way back when, one of the parts of the cell theory was that all cells come from existing cells. So in order for you to get more cells, they have to divide. Hence us talking about mitosis today. And that goes for any living thing on Earth, whether it's a prokaryote or eukaryote, cells divide to make more cells. If we're talking about prokaryotes, remember, prokaryotes are bacteria. The division for them, well, first of all, they don't have a nucleus, so they have their own special kind of division that's called binary fission. Because technically, mitosis is the division of the nucleus. And since bacteria don't have a nucleus, their division is called binary fission. They have a fairly small chromosome, and they can copy their chromosome and divide into two, splitting the copied chromosomes, dividing them into each of the daughter cells in roughly 20 minutes. So bacteria can divide very, very rapidly. In eukaryotes, the genome is a little bit more complex, so it is definitely more time-consuming and takes hours, not minutes. This division for eukaryotes is called mitosis. And I just wanted to remind you what eukaryotes are. Eukaryotes are animals, plants, fungi, and protists. So essentially all of the kingdoms other than bacteria. Now again, as a reminder, DNA is copied before cell division. It's copied using enzymes, and those enzymes were helicase, DNA polymerase, and ligase. DNA is copied so that during cell division, both daughter cells get a full identical copy of DNA. So after replication occurs, the DNA inside the cell is going to condense to what are called chromosomes. So we're going to kind of talk a little bit more about what the heck a chromosome is right now. If you were to actually just see the DNA inside the cell, after it's copied, there are two of them, and If this DNA were to be condensed, we could make a chromosome. So imagine two identical chromosomes after replication. And so what the cell does is it actually attaches those two chromosomes together to a structure that looks like this. So they show that in this diagram here. If this is the chromosome, after copying, we have two chromosomes that are identical, and we call that a duplicated chromosome. And they are held together by a structure called the centromere. Because the chromosomes are identical, we call them, we have a special name for them. They're called sister chromatids. Sister chromatids means identical chromosomes. And the whole point of mitosis is to pull these identical chromosomes apart, these two sister chromatids, and put each one into one of the two daughter cells that's going to form at the end of cell division. So going back up here, after replication, each chromosome consists of two identical sister chromatids attached at a centromere. We are going to call this either a replicated or a duplicated chromosome. Either one is fine. It just depends upon the source. I tend to say replicated chromosome. Your textbook tends to say duplicated chromosome. Either is fine. And a replicated chromosome is actually two identical chromosomes held together, attached at the centromere. When the cell divides, the sister chromatids will separate from one another or each other. One chromatid goes to each of the two new cells. Now, cell division, and actually to be real clear, mitosis is actually nuclear division. Mitosis occurs in steps: prophase, metaphase, anaphase, and telophase. In middle school, you might have learned prometaphase, but we're just going to leave that out for now, and we're just going to stick with the big four: prophase, metaphase, anaphase, and telophase. So, cell division begins by the DNA, which is actually in the form of chromatin, is going to condense to replicated chromosomes, which are shown here. The nuclear membrane is going to disappear or dissolve. And so I can show that typically by showing a dotted line going around the nuclear membrane, just to show that it's going to go away. And centrioles and spindle fibers appear in animal cells. Those are the organelles that are going to be used to pull the sister chromatids apart, the replicated chromosomes apart in the cell division. So we have prophase comes first, metaphase comes second. Metaphase is where the sister chromatids or replicated chromosomes line up along the center of the cell. And you can see that here. The spindle fibers attach at the centromeres. So where every centromere is, a spindle fiber will attach. So I think M for middle. Anaphase comes next, and I think A for away. In anaphase, the sister chromatids split, and the chromosomes move toward the ends of the cell, the opposite ends of the cell. So they're going to be pulled apart. So if you've wondered why the heck do they look like kind of like these C shapes, it's because if you take now an individual chromosome and you begin to pull it that way, it's going to flop over this way. So they tend to look like that as they're being pulled. So we had prophase, we had metaphase, we have anaphase, and then we have telophase. And in a way, telophase is like the opposite of prophase. The chromosomes are going to unwind back to chromatin. The nuclear membrane reappears. The centrioles begin to disappear. So the nuclear membrane you can see is returning. The centrioles are going to start to go away. The chromosomes are going to begin to unwind back to chromatin. And then you're also going to get a pinching in of the cell, if it's an animal cell. If it's a plant cell, you're not going to get the pinching in. You're going to get something else. I'll talk about that in just a second. So, prophase, metaphase, anaphase, and telophase, PMAT, are all parts of the mitosis or the M phase of the cell cycle. Then you have cytokinesis. Cytokinesis is its own phase, separate from mitosis, and it is defined as the separation of the cytoplasm, where the cell finally pinches into two or separates into two. If you have an animal cell, you are going to get that pinching. And that pinch is called a cleavage furrow in an animal, like I mentioned up here. Plant cells have a cell wall, so they can't pinch in. So what they do is they form a new cell wall down the middle to divide the two new cells, and that's called a cell plate. So animal cells pinch in at what's called the cleavage furrow. And plant cells will build a new cell wall in between the two daughter cells, and that is called the cell plate. By the way, the two cells that are formed are called daughter cells. So when a cell is not dividing, it is in a phase called interphase. Interphase is going to take up much of a cell's life. It's not constantly dividing. Typically, the cell is just doing its normal daily job. So interphase is what a cell typically is going to be found in. The cell is doing its normal daily job, and at a certain point it might grow so big that it triggers the cell to copy its DNA in preparation for cell division. But that DNA replication is going to happen in interphase. If you want to know what a cell looks like in interphase, it looks like these cells here. This one, this one, this one, and this one. You're going to see a visible nucleus. You are not going to see chromosomes because the DNA is in the form of chromatin inside the nucleus. Now, replication might occur during this phase, and it happens in a subset of interphase called the S phase, and I'm going to come back to that in just a second. So you can think of the cell's life or life cycle looking somewhat like a clock. And this is called the cell cycle. The cell has its main phase, interphase. It has the mitotic or mitosis phase, and it has cytokinesis. So this all three of these were in a reading that I had you do, or will have you do. Those are the three main phases of the cell's life. In mitosis, as we talked about before, you just need to know prophase, metaphase, anaphase, and telophase. And in interphase, there are three subsets of interphase, G1, S, and G2. I will talk about those down here. But first, let's just answer the question, why do cells divide? Why even undergo mitosis at all? Well, you need to grow. You need more cells in order to do that. So you have to divide, your cells have to divide so that you can get bigger. If you have any damage, you need to repair or replace damaged cells. So cell division helps that happen. And then finally, we talked about cells want to stay small. If they get too big, their cell surface area to volume ratio becomes too much volume and too little surface area to sustain the cell. So that is also going to be a trigger for the cell to divide. So our last little bit here is to reduce cell size. Now going back to interphase, the three subset phases of interphase are G1, S, and G2. In the G1 phase, the cells will grow, they carry out their normal functions, and they're just doing their daily job. There is an offshoot of the G1 phase called the G0, and not all cells will actually go into the G0, but some cells do. G0 means the cell might not divide again. It might live out its life until it dies and never divide ever in the future. Examples in the body are heart muscle cells that are multinucleated and many neurons. They do not divide ever again. Neurons are in your nervous system. Think your brain and your spinal cord and your peripheral nerves. Now, G0 is not typical. Most cells do divide, and so if they get ready to divide, they have to copy their DNA, and that's going to happen in what's called the S phase. So DNA replication will occur in the S phase. After DNA replication occurs, we enter the final subset of interphase called G2, where you have some additional growth, you have preparations for division, and You also make organelle, extra organelles, so you can have organelle division as well. And then the cell would enter the M phase, which, as you guys know, would be mitosis. So, to recap, you're going to want to know the difference between division between prokaryotes and eukaryotes. You are going to want to understand what a chromosome is, what sister chromatids are and how they make up a replicated chromosome. How do you get the copies of this replicated chromosome? Well, it happens in DNA replication and interphase. The point of having cell division is to pull those copies apart so that each of those copied chromosomes, one can end up in each of the two daughter cells. You're going to want to know the phases, prophase, metaphase, anaphase, and telophase, and know what chromosomal changes occur in each of the phases. Cytokinesis is the division of the cytoplasm and how animals and plants differ. What interphase is, what the cell is doing in interphase. And that's it. Oh, and then finally, why cells divide. So why is it important that cells need to divide?